welcome back to the next video. It is about gel electrophoresis. So what is this gel electrophoresis and why is it done? I hope you remember the first few steps that we covered while understanding all the steps under biotechnology. The first thing that we did under biotechnology is identifying the gene of interest. This one. Where was it? It was probably inside one bacteria or any organism. So we cannot take just that gene out. So we extracted the entire DNA like so. This entire thing we extracted. Once we extracted that, what did we do? We cut it using restriction enzymes. This part, restriction enzyme were used and then we cut it out. Now we have a mixture of genome with us. All right. I have, let's say in a test tube, I'll take a test tube. It is easy for me to take a test tube than something else. So I am going to take a test tube in which I have my entire genome, and in this one part somewhere is my sorry, yeah, somewhere is my gene of interest. All right. So what do? I pour this entire thing into a gel electrophoresis unit, which as you can see here is a unit consisting of many components. All right. This unit has here a matrix. It is called as a matrix. This part, the gray color part that you can see, the jelly substance made up of agarose. What is agarose? It is a polysaccharide extracted from seaweed. This is your 1 mark question, MCQ probably. Where is it extracted from? Seaweed. What are we talking about? We are talking about the matrix in which we are going to do the entire electrophoresis process. All right. So this matrix is made up of agarose, polysaccharide it is, agarose gel is filled in this unit. This agarose is extracted from seaweeds. Please remember that. Okay. Now, in the agarose, there are tiny wells. As you can see here, there are tiny wells. Into this wells, I'm going to add my genome having the gene of interest. Okay. So where am I adding? I'm adding it here into well number 2, 3 and 4. Why am I not adding in well number 1? Into well number 1, I would be adding a same set of genome which I extract, that is I extract DNA from the same organism. However, I do not cut it using restriction enzyme. So what is the difference between well number 1 and 2, 3, 4? Inside 2, 3, 4, this and this, you have the DNA extracted, the entire genome which is cut by restriction enzymes, poured in this and this. In well number 1, on the other hand, I add DNA extracted from the same organism, however, not cut by restriction enzyme. Now, what do I do next? I connect this unit to electricity, power. Okay. I connect this side and this side to electricity. How do I connect? I connect in such a way that the side where wells are present, I connect to the negative terminal, and other end, I connect to the positive terminal. All right. So as you know, DNA is negatively charged. This much you know. DNA being negatively charged will be repelled from this end and will start moving towards the other end. Clear? So how does it move? It moves in such a way that the largest fragment will take more time and the smallest fragment will take less time. So the smallest fragment will be ahead, in front in the gel, and the largest will be left behind. So from this, I should know the length of my gene of interest. Otherwise, I cannot understand from this where is my gene of interest. All right. So, depending on the length of my gene of interest, I need to identify it. For that, there are certain other criteria. While adding the DNA into the wells, you are also going to add a dye. The name of the dye is ETBR, or ethidium bromide. Name of the dye is ethidium bromide. Once you add ethidium bromide and the gel has started running, at some point you stop it and then observe this under UV light. Under UV light. Only then you will be able to see orange bands. orange colored bands. From this, you will identify where all are the fragments of DNA. Why are they in fragmented form? It is because it is cut with the restriction enzyme. However, in the first well, you will see one large band instead of tiny fragments. The reason being, it is uncut, right? It is undigested by the restriction enzyme. I hope this process is clear. Thank you.